Dear student, welcome to the Medical Genetic course. Today's lesson is cell division. By the end of this video, you will be able to define the cell cycle, purposes, and stages. Identify the stages and phases of mitosis. The content of this lecture includes the cell cycle, purpose, and stages. It includes interphase and M phase. Second, the stages of mitosis. And lastly, the phases of meiosis. I will start with a question. Why is cell division so important? And what is the purpose of it? Well, at conception, the human zygote consists of a single cell. This undergoes rapid division, leading ultimately to a mature human. Also, most organs and tissues in our bodies, such as bone marrow and skin cells, continue to divide throughout life. This process of somatic cell division is known mitosis. Cell division remains important in normal cell turnover, such as our skin and gut, where cells are continuously renewed. Another type of cell division is meiosis, which is required for germ cell division. When an egg cell and sperm cell unite to form a zygote, their chromosomes are combined into a single cell. Because humans are diploid organisms, meiosis is the mechanism to reduce the number of chromosomes in gametes to the haploid state. Otherwise, the zygote would have 92 chromosomes. All cells in our body have a cell cycle. In rapidly dividing cells, this lasts for between 16 and 24 hours. Cell cycle is divided into interphase and M phase. Interphase is the largest portion of cell cycle. However, M phase or mitosis is the phase where a cell divides to become two daughter cells. Cells that have stopped dividing, such as neurons, usually arrest in this phase and entered a non-cyclic stage known as G0. Interphase is very variable in length and is responsible for the variation in generation time between different cell populations. Cells that have stopped dividing, such as neuron, usually arrest in this phase and are said to have entered a non-cyclic stage known as G0. The purpose of interphase in a cell type is grow, perform cellular function, cell division by DNA synthesis. A typical cell spends most of its life in interphase. This portion of cell cycle is divided into three phases: G1, S, which stands for synthesis, and G2, or gap, is the growth phase, whereas synthesis of RNA and protein takes place. DNA replication occurs during the S phase. By the end of this phase, cell contains two identical copies of each of the 46 chromosomes. G2 phase, the cell continues to grow and prepares for division. The cell double-checks the duplicated chromosomes for error, making any needed repairs. Once G2 ends, the cell is ready to proceed into mitosis. Before the cell enters mitosis, for example, DNA replication must be accurate and complete. A checkpoint is a surveillance mechanism that stops the progress of the cell cycle if there is any chromosomal DNA damage, or if there is certain critical process such as DNA replication during S phase or chromosomal alignment during M phase have not been properly completed. Cancer is the uncontrolled division of cells, which results from an improperly regulated cell cycle. After the cell contents and chromosomes replicate in the interphase, cells stop growing and mitosis begins. During mitosis, the duplicated chromosome separates from each other, result in two identical sets of chromosome. Each set of chromosome will become a part of a new daughter cell. There are 5 stages in mitosis or M phase. The first phase of mitosis is prophase, in which nuclear membrane starts to go away. Centromere migrate into the opposite side of the cells and chromosomes start to get condensed. The second stage is prometaphase, in which nuclear membrane is gone. Chromosomes are lining up in the middle, and there will be extension of microtubules from centromere toward chromosome. The third stage is metaphase, where microtubules pull up chromatid kinetochore, which is the site of attachment of the chromosome to the dynamic microtubules of the mitotic spindle. Chromosomes reach the most condensed state and are aligned in the middle of the cell, as you see in the figure. The spindle fiber attached to the centromere begin to shorten. Karyotype analyses are usually made in this stage. In anaphase stage, spindle fibers shorten, causing the centromere to split. Each sister chromatid is pulled to the opposite end of the cell, result in a single cell within 92 distinct chromatids. 46 of those chromatids are moving toward one end of the cell, and 46 are moving toward the opposite end. The last stage is telophase, in which the mitotic spindle deassembles and the nuclear envelope reforms. Lastly, daughter cells are created by equal division of the parent cell's cytoplasm through a process called cytokinesis. Cytokinesis immediately follows mitosis and precedes G1. The second type of cell division is meiosis. It's the process of nuclear division that occurred during the final stage of gamete formation. When an egg cell and sperm cell unite to form a zygote, their chromosomes are combined into a single cell. Because humans are diploid organisms, there must be a mechanism to reduce the number of chromosomes in gametes to the haploid state. Otherwise, the zygote would have 92 instead of normal 46 chromosomes. Therefore, the primary mechanism by which haploid gametes are formed from diploid precursor cells is meiosis. Meiosis has two phases: meiosis 1, that is called reduction division, and meiosis 2, which is responsible for producing haploid gametes. I'm going to highlight the important events that occur during meiosis. First step is prophase 1. Genetic recombination occurs, also called meiotic crossing over. This happens when one pair of chromosomes, homologous segment of DNA, are exchanged between non-sister chromatid of each pair of homologous chromosomes as shown in the picture, thus ensuring that none of the gametes produced by meiosis will be identical to another. This independent assortment allows maternal and paternal chromosome to become shuffled during formation of the gametes. After prophase, other stages including metaphase, anaphase, and telophase occur and end up with two cells that are genetically different. The second cell division in meiosis is meiosis 2. It's the same as an ordinary mitotic division. Each chromosome, which exists as a pair of chromatid, become aligned along the equatorial plane and then splits, leading to the formation of two new daughter gametes known as spermatids or ova. There are medical consequences of chromosomal non-disjunction. Meiotic mistakes appear to be common. Homologous chromosome may fail to separate and is called non-disjunction. It could occur during meiosis 1, or sister chromatids may fail to come apart during meiosis 2. When one of these situations occurs, gametes are formed to contain an abnormal number of chromosomes. We will talk about it in more details in chromosomal abnormalities lecture. Failure to recombine properly can lead to chromosomal missegregation, which we call non-disjunction. It may cause pregnancy loss or abnormal karyotype such as Down syndrome. At the end, these are the references for the lecture. You can always go back and read more on the topic. In summary, we have talked about the cell cycle, purpose, and stages. We also talked about the stages of mitosis and the phases of meiosis.